Hi, I'm Dr. Thomas Martin, and we're here in Scotland at Myeloma 2017. I'm here with my colleagues Dr. Boyce, Dr. Driessen, and Dr. Mitsiades, and we just finished a session on novel therapeutics in multiple myeloma. It was a great session. I'll start with you. Dr. Boyce, what did you conclude from your session today?
Today we talked about how it's been very exciting times for those of us who've worked on BCL2 proteins, because these are now targetable with drugs like Venetoclax, and looked at how its activity in myeloma can be quite effective in a subgroup of myelomas, and is real promising also in combinations, and looking forward to the future of using another inhibitor in this class, the MCL1 inhibitor.
Thank you. And Dr. Driessen, tell me what you concluded from your session today.
So we've been working on Nelfinavir, which, unlike most other novel drugs in myeloma, is an old drug. It's an HIV drug. It's an approved drug that's available to patients. And the aim really is to find a therapy for patients that are multi-refractory with myeloma and with an available treatment. And we have presented at last ASH a combination of Bortezomib, Dexamethasone, and Nelfinavir in heavily pretreated patients, where we have seen something like the highest response rate that has been seen in this type of patient population. And the real question now is it real, and what is the mechanism and what is the target? And in the session today, I digged in some in vitro data we have that show that indeed Nelfinavir is an inhibitor of intracellular protein transport, which explains how Nelfinavir sensitizes myeloma cells, in particular refractory myeloma cells, to proteasome inhibitor treatment. So I think we do have a biological mechanism now that can be understood on the basis of plasma cell biology. And the other important take home, I think, from this session for me was that there were also other investigators like the Little Rock group that have meanwhile tried this available treatment, Bortezomib, Dexamethasone with Nelfinavir, in myeloma patients out of clinical trials in multi-refractory settings, and anecdotal evidence they report that they see similar great responses as we've seen in the study. I think that's going to support this concept into the future very much.
So now for both of you, there is these combinations of Velcade plus Dexamethasone in either Nelfinavir or Venetoclax. Is in your practice or in your experience, are there patients that we should be thinking of one of these drugs versus the other for?
I think the mechanisms of action of the two different drugs are quite different, and I think we have to consider that. I think the data with Venetoclax in the combination would be, it's going to enhance the activity of Bortezomib. Primarily when Bortezomib has some activity, I think it would be a little bit different in the case of Nelfinavir, where I would say it's more about when you have resistance to Bortezomib or to other drugs, probably because of the pumps.
So for Nelfinavir, it's increasing the activity of proteasome inhibitors, whether cells are sensitive or resistant to proteasome inhibitors. So it would make sense to use it also in non-resistant myeloma, in my view. But I think the important point for clinical practice really is that, of course, Nelfinavir is an approved drug that's available, and so are Bortezomib, Dexamethasone. So we are looking at something that has a monthly therapy price of something like, I don't know, $1,000 or less in the generic state. When we are looking at next generation combinations that are in most countries, in most settings, even not available, and have costs about 20, 30-fold. That I think this is also something to consider, and I think it's a broadly applicable concept, really, that doesn't depend really on certain types of cytogenetics like Venetoclax in a way does, and importantly, it's available to patients.
And I think, Dr. Boyce, you and the people from Emory have really beautifully shown that specifically in the translocation 11-14 population, that they are enhanced at having overexpression of the BCL2 pathway. And perhaps that group of patients may be more sensitive to this therapeutic. Is that correct?
Right. I think the way moving forward with Venetoclax would be as a single agent, or with Dexamethasone, where we've shown that there's enhanced activity in the patients who have the 11-14 translocation, that there's those are highly active. I think in the other patients, combinations with Bortezomib work. So the data from the trial with Bortezomib and Dexamethasone and Venetoclax showed that, we think about in a patient population where the response rate would have been about 60% with Bortezomib and Dexamethasone, it went up to almost 100% when you added Venetoclax in that population.
And for Nelfinavir, so really the biomarker that what you presented really nicely today was that people that have proteasome inhibitor resistance, although you say now, which I think is great, that even in sensitive disease it probably would work well, but specifically for proteasome inhibitor resistance, that this is a great drug to use. Is there any other biomarker that you have found or are looking for?
So I think it makes a difference what type of resistance you're looking at, and it especially makes sense really for carfilzomib resistance, because as we have seen, carfilzomib resistance is mostly mediated by upregulation of MDR pumps. So if you were to look at the activity of MDR pumps on myeloma cells, that would be a good biomarker to predict an additional positive effect of drugs like Nelfinavir and others.
And I think what came up at this session today too is anthracyclines was another. Resistance to anthracyclines may be augmented or maybe reversed with the use of Nelfinavir. Is that correct?
So Nelfinavir is inhibiting a broad number of these MDR pumps, including PGP, and good substrates of PGP that are being used in myeloma are daunorubicin, are panobinostat, is carfilzomib, and also the novel epoxyketone proteasome inhibitors. Poor substrates for ABCB1 are, for example, cyclophosphamide. So that's also something to consider. And I think we have lost the issue of MDR pumps out of sight a little bit in the myeloma field. We do know that they play a role. We know it from the old days when we used chemotherapy, that they are very effectively pumping out our drugs out of the cells. And I think that's maybe a point to be revisited these days.
And Dr. Mitsiades, so your session today was really looking at the next generation of drugs or pathways that we can target. So tell us what you found and what you presented.
So we presented extensive ongoing work of our team and collaborators on identifying what are the genes that are essential for myeloma cells, and comparing that with other malignancies in order to be able to tease out what's really very special about multiple myeloma. And in the process of doing that, we were very reassured about findings that were consistent with prior knowledge or findings that make a lot of clinical sense. For instance, Icaros and Aiolos, two key transcription factors that are degraded by the thalidomide derivatives, come up as genes that are preferentially important for myeloma compared to all other cancers that we're able to analyze. And so that gives us a sense perhaps as to why this class of drugs has this level of activity in multiple myeloma. But beyond those two genes, there were several other transcription factors, epigenetic regulators, some kinases that regulate NF-kappa B activity, and other candidate genes that we've identified. And probably the strongest signal was a signal that was related to transcription factors, cofactors, and epigenetic regulators that seem to play an important role in multiple myeloma. Some of them may play a role in other blood cancers or may play a role in subsets of solid tumors, but it was very intriguing to see this a large number of transcription factors and transcriptional regulators coming up. This is not necessarily something that can be easily targeted therapeutically. Some of the epigenetic regulators have small molecule inhibitors that are available, and some of them are in active preclinical investigation. But I would say the most exciting piece coming out of this work is also the realization that some important targets in this disease may not necessarily be the targets that are mutated or expressed at very high levels. Sometimes the myeloma tumor cells may depend on targets that we have overlooked for many years because we couldn't find them in gene expression profiling studies about what is overexpressed in tumor versus normal, or we haven't necessarily seen these molecules coming up in lists of recurrently mutated genes. And so that means that we have an array of new promising targets for which we may not necessarily have drugs right now, but we could in the near future. And because it appears that these targets are so preferentially important for myeloma versus other lineages, perhaps they will be able to recapitulate for myeloma some of the activity that has been observed with thalidomide derivatives or other drugs that target the plasma cell biology of myeloma, even if they don't necessarily target the oncogenic events that make those plasma cells malignant.
And you mentioned also a little bit about super enhancers, and we have some drugs that perhaps are going to target super enhancers. Do you think that's going to be an important way for us to provide anti-myeloma effects?
You know, it's a complex topic, and it's clear that some very important genes in myeloma are driven by super enhancers. But it's also clear from our experience that there are many other genes that are very important for myeloma, and perhaps more so important for myeloma than other cancers, that we cannot identify them to be near to a super enhancer. And there's no evidence for us to suggest that some super enhancer is the real drive for expression of those genes. And so that tells us is that maybe there's a lot more to regulation of the transcriptome in myeloma than super enhancers. Or perhaps that the definition of how to identify super enhancer may have to be modified, and perhaps one of the values of this research effort that we and our collaborators are undertaking is that by providing these functional data, we can now perhaps rederive better definitions of super enhancers, and that can provide potentially better markers for identification of genes that are interesting, or even for potential future clinical practice tests that perhaps can really tell us what are the most important super enhancers. And so this is an ongoing work, but clearly something that's quite exciting and can yield potentially very important biological and translational insights.
Great. Well, thank you very much. We're from Myeloma 2017. I'll summarize by saying we've had a lot of riches in myeloma therapeutics. We have some blockbuster drugs, including monoclonal antibodies, proteasome inhibitors, and immunomodulatory drugs. But now we have two drugs that we talked about today, Venetoclax, that potentially can work in patients that have overexpression of the BCL2 pathway. And there might be a novel biomarker. The 1114 translocation patients may have a higher sensitivity to these drugs. And there's currently a phase 3 trial testing, in fact, Velcade dexamethasone with or without Venetoclax in patients with relapsed myeloma with 1 to 3 prior lines of therapy, and not specifically only in the 1114, so we'll see how that works out. We also have another drug, Nelfinavir, that can work in patients that have carfilzomib refractoriness, or maybe refractory to other drugs that utilize MDR for part of its resistant mechanism. And so it might actually make cells more sensitive to carfilzomib or other proteasome inhibitors, or to anthracyclines, which is great. We're trying to divide myeloma now into many different topics, or many different types of myeloma. And Dr. Mitsiades and others are looking at a variety of pathways, including with transcription factors and with super enhancers, to see if we can actually target other pathways that may be successful in the future. Thank you very much.